So now let us study the second type of gametogenesis, that is oogenesis. We have to study about oogenesis. Name itself indicates double O. This word stands for ovum or egg cell. Ovum or egg cell, that is female gamete, and genesis stands for formation. So the meaning is very simple. Oogenesis, it is nothing but the process of formation of female gametes. Process of formation of female gametes, that is ova or egg cells. That is ova or egg cells. Now let us talk about the actual process of oogenesis. All of you are known that oogenesis, that is formation ova, formation of egg cells, it takes place in the female gonads, that is ovaries. Actually, the process of oogenesis is considered as highly lengthy and very wasteful process. What did I say? The process of oogenesis is considered as highly lengthy and very much wasteful process. Highly lengthy. Why this is so? Because we have already discussed in human female the process of oogenesis, process of formation of ova, it starts before the birth of female baby. Before the birth of female baby. It means the oogenesis starts in the ovary of that female fetus. In the ovary of that female fetus before birth, and it completes only during fertilization. Better to say during fertilization. Means whenever the sperm penetrates the developing stage of ovum, at that time only the actual oogenesis get finished, actual ovum is formed. It means that we can say that process of oogenesis is very much lengthy, and it is highly wasteful process also. Wasteful. Why this is so? Because while discussing the histological structure of ovary, I have taught you that in each ovary during oogenesis about 2 millions of primordial follicles are formed. Millions and millions of oogonia are formed. 2 millions of, we can say, primordial follicles are formed, and actually in the reproductive life of that female, the egg form, the number of eggs formed, the number of ova formed are more or less 400, or we can say 420. And that's why this process is lengthy and wasteful process. Now we have to study the different phases of oogenesis. आता आपल्याला oogenesisच्या phases कोणत्या आहेत याचा अभ्यास करायचा आहे. Actually, the phases involved during oogenesis are similar to that of phases involved during spermatogenesis. आपण spermatogenesis शिकत असताना ज्या 3 phases अभ्यास केला त्याच 3 phases या ठिकाणी सुद्धा आहेत oogenesis मध्ये with slight difference. दोघांमध्ये थोडासा फरक आहे. But the basic phases, basic steps involved during spermatogenesis and oogenesis are similar. So which are those 3 phases? You know, the first phase is called phase of multiplication. Second phase, phase of growth, and third phase, phase of maturation. So there are 3 phases here in oogenesis also, such as first multiplication phase, second growth phase, and third maturation phase. So now let us start the study of that phases one by one each. The first phase is multiplication phase. First multiplication phase. I said just now that the process of oogenesis, it starts in the ovary of female fetus. Now, some of the germinal epithelial cells of the ovary undergo mitotic repeated cell division. Listen carefully. During oogenesis, initially some of the germinal epithelial cells which form the lining of ovary undergo repeated mitotic cell division, repeated mitotic cell division to form numerous very small and diploid cells known as oogonia. Let me explain you properly. While studying histological structure of ovary, we have already studied. आपण फिमेल रिप्रोडक्टिव्ह सिस्टीम शिकत असताना ओव्हरीची हिस्टॉलॉजिकल स्ट्रक्चर शिकत असताना असं शिकलो की सपोज हियर इज ovary. You just imagine, and you know that ovary is internally lined by a single layer of cuboidal epithelial cells. It is known as germinal epithelium, isn't it? Germinal epithelium. These germinal epithelial cells are diploid in nature, that is, they have two sets of chromosome, means that each germinal epithelial cell contains 23 pairs of chromosome, 46 chromosome. अर्थात ओव्हरीचं लाइनिंग तयार करणाऱ्या या ज्या जर्मिनल एपिथेलियल सेल्स आहे ही प्रत्येक सेल डिप्लॉइड असते. प्रत्येक सेल मध्ये गुणसूत्रांच्या 23 जोड्या असतात किंवा 46 गुणसूत्र असतात आणि या जर्मिनल एपिथेलियल सेल्सला आपण प्रायमॉर्डियल जर्म सेल्स असं सुद्धा म्हणतो. आता मी असं म्हटलं की ड्यूरिंग oogenesis होत असताना सुरुवातीला some of the germinal epithelial cells, not all, some of the germinal epithelial cells which are comparatively larger in size as well as have more distinct nucleus, having more prominent nucleus, such germinal epithelial cells undergo repeated mitotic cell divisions. Remember, mitotic division, mitotic in the sense equational division. You know, mitosis or equational division means a type of cell division in which the parent cell produces daughter cells receiving equal number of chromosome, same number of chromosome. अर्थात आता जसं मी म्हटलं की या जर्मिनल एपिथेलियल सेल्स डिप्लॉइड आहेत, मी या ठिकाणी शॉर्टकट असं लिहिलं, these are germinal epithelial cells. आणि या जर्मिनल एपिथेलियल सेल्स डिप्लॉइड आहेत, त्यांचं मायटोसिस या पद्धतीने विभाजन होत जातं आणि त्यांच्यापासून असंख्य लहान लहान सेल्स तयार होतात. Numerous number of very small cells are formed. That small cells are called as oogonia, or may be called as the egg mother cells. Obviously, these oogonia are also diploid in nature. Now, we have already studied that these oogonia protrude in the form of small cord-like structure into the stroma of ovary. This cord is called as egg tube of flagell. We have already studied egg tube of flagell. And at the end of this egg tube of flagell, there is a formation of such rounded mass of oogonia. It is known as egg nest. Now the same thing here I am going to draw here. I am going to explain. Just imagine this much part of germinal epithelium here. I am going to draw. Look, this is supposed to be the germinal epithelium. Okay, germinal epithelial cells which are diploid in nature, and I said the germinal epithelial cells undergo repeated mitotic cell divisions to form many diploid cells. You know, these diploid cells are called as the oogonia. This phase is nothing but the multiplication phase. For now, in this one egg nest, as there are presence of many oogonia, एका एग नेस्ट मध्ये असंख्य oogonia आहेत आणि अर्थात यापैकी सिंगल ओगोनियम undergoes differentiation. सिंगल ओगोनियम undergoes differentiation. अर्थात या असंख्य oogonia ज्या आहेत त्यापैकी एक ओगोनियल सेलचं डिफरेंशिएशन होतं. It means that single oogonium appears structurally and physiologically different than remaining oogonial cells. इतर ओगोनियल सेल्स पेक्षा एक फक्त ओगोनियल सेलचं डिफरेंशिएशन होतं and that differentiated oogonial cell acts as a primary oocyte. So here we have seen about the multiplication phase. I have already explained you. In this egg nest, only one oogonial cell undergoes differentiation and it acts as the primary oocyte, and remaining oogonial cell surround that primary oocyte to form a structure called as primordial follicle. Of course, this primary oocyte is also diploid in nature. अर्थात ही पण diploidच आहे. It means here occurs the multiplication phase. आता हे जे primary oocyte आहे, this primary oocyte it increases in its size. It means growth occurs. Due to the accumulation of cytoplasm, different cell organelles, DNA, etc. and reserve food material, little bit reserve food material. This is called as the growth phase. याला आपण growth phase असं म्हणतो. Actually, the growth phase is extended maybe up to 12 to 13 years. Now this primary oocyte enclosed within that developing follicle, primordial follicle, then primary follicle, then secondary follicle. Here, this primary oocyte starts its maturation division. अर्थात हे जे प्रायमरी oocyte आहे, आता हे प्रायमरी oocyte आपण असं म्हणूया की सुरुवातीला हे प्रायमरी oocyte प्रेझेंट असतं प्रायमॉर्डियल फॉलिकल मध्ये, त्यानंतर इट इज प्रेझेंट in secondary follicle because we know that the primordial follicle undergoes modification to form primary follicle. Primary follicle मध्ये modification होतं, काय होतं ते आपण मागे शिकलो. Primary follicle चं रूपांतर होतं secondary follicle मध्ये, and you know all these developing follicles enclose the developmental stage of ovum, that is primary oocyte. Actually this primary oocyte is future ovum. Listen, प्रायमरी oocyte हे फ्युचर ओवम आहे, कारण प्रायमरी oocyte पासूनच खरखुरं एग सेल, खरीखुरं फिमेल गॅमेट, खरखुरं ओवम तयार होणार असतं. अर्थात स्टिल that primary oocyte is enclosed within the follicle. Here I have shown primordial, primary, secondary, and now when it attains the sexual maturity, that is we can say around the age of 12 or 13 years, what happens? This primary oocyte is converted into secondary oocyte. How is that? Let me explain you. Actually, before attaining puberty येण्याच्या आधी, sexual maturity येण्याच्या आधी, हे जे प्रायमरी oocyte इथे प्रायमॉर्डियल फॉलिकल मध्ये असतं, ते त्याचा मॅच्युरेशन डिव्हिजन सुरू करतं. Means this primary oocyte, it starts its maturation division. It starts its first maturation division just like the primary spermatocyte in spermatogenesis, and you know this first maturation division is nothing but the meiosis first, and it is reductional division. आपल्या सगळ्यांना माहित आहे जे स्पर्मॅटोजेनेसिस मध्ये होतं त्याच प्रकारे. मग या ठिकाणी हा प्रायमरी oocyteचं फर्स्ट मॅच्युरेशन डिव्हिजन सुरू झालेलं असतं when it is present in primordial follicle, but that first maturation division, meiosis first, does not complete. ते पूर्ण होत नाही. First maturation division, that is meiosis first, is arrested, is stopped in prophase first of meiosis first. Prophase first of meiosis first. याचा अर्थ ह्या प्रायमरी oocyteचं फर्स्ट मॅच्युरेशन डिव्हिजन सुरू झालेलं असतं पण ते पूर्ण होत नाही. ते या ठिकाणी थोपवलं जातं, या ठिकाणी थांबवलं जातं आणि प्रायमरी oocyte या स्टेजला येऊन तसंच थांबतं. कुठपर्यंत? अप टू द अटेनमेंट ऑफ प्युबर्टी, वय वर्ष 12-13 होईपर्यंत. आणि याला म्हटलं जातं फर्स्ट रेस्टिंग पिरियड. म्हणजे हा प्रायमरी oocyte जवळपास 12 ते 13 वर्ष जणू रेस्टिंग पिरियड मध्ये असतो, फर्स्ट रेस्टिंग पिरियड मध्ये असतो. नाउ आफ्टर अटेनिंग प्युबर्टी, प्युबर्टी आल्यानंतर नाउ दिस प्रायमरी oocyte completes its first maturation division. प्युबर्टी आल्यानंतर या प्रायमरी oocyteचं फर्स्ट मॅच्युरेशन डिव्हिजन पूर्ण होतं. फर्स्ट मॅच्युरेशन डिव्हिजन अर्थात meiosis first, and ultimately there are formation of two haploid cells. First maturation division, that is nothing but the meiosis first, and you know it is reductional division. Means what? Chromosomal number reduces to half. Chromosomal number half झाला. आपल्याला माहित आहे या ठिकाणी प्रायमरी oocyte डिप्लॉइड आहे आणि त्याचं मियोसिस झाल्यामुळे, रिडक्शनल डिव्हिजन झाल्यामुळे 2 हॅप्लॉइड सेल्स तयार झाल्या. बट दिस डिव्हिजन इज हायली अनइक्वल. The larger cell, one is very large cell, one is very small cell. Both are haploid, no problem. Both are haploid, but one is very large, one is very small. The larger cell is known as secondary oocyte. Larger cell जी आहे ती आहे secondary oocyte, and smaller cell is called first polar body. First polar body, or it may be called as the polocyte. First polar body or polocyte. This first maturation division and the formation of secondary oocyte takes place within the Graafian follicle, or exactly at the time of ovulation. अर्थात प्रायमरी oocyteचं फर्स्ट मॅच्युरेशन डिव्हिजन पूर्ण होऊन सेकंडरी oocyte तयार होऊन एका बाजूला फर्स्ट पोलर बॉडी तयार होणं, ही प्रक्रिया होत असते या ठिकाणी ग्राफियन फॉलिकल मध्ये किंवा एक्झॅक्टली ॲट द टाइम ऑफ ओव्ह्युलेशन. यावेळेला ओव्ह्युलेशन होतं त्यावेळेला. नाउ this secondary oocyte is largest because it almost receive entire cytoplasm and entire content of primary oocyte. प्रायमरी oocyteचं जवळपास सगळं सायटोप्लाझम, सगळं कंटेंट याच्यात आलेलं असतं सेकंडरी oocyte मध्ये. म्हणून सेकंडरी oocyte is very much large, and this first polar body receive a very little cytoplasm, and therefore it appears very small, but the chromosomal number is same. Both are haploid. Now this secondary oocyte, you know, shaded outside the ovary, you know, it is pushed into the oviduct, ampulla part of oviduct, and here this secondary oocyte remains as it is up to the, we can say, approach of sperm. When the sperm reaches this secondary oocyte, at that time this secondary oocyte undergoes second maturation division. This second maturation division is nothing but the meiosis second. अर्थात याला आपण meiosis second म्हणतो. आणि meiosis second आपल्याला माहिती आहे it is equational division. म्हणजे या ठिकाणी आता गुणसूत्रांची संख्या निम्मी होणार नाहीये, गुणसूत्रांची संख्या स्थिर राहणार आहे. आणि ज्या वेळेला या सेकंडरी oocyteला स्पर्म पेनिट्रेट करतो, त्यावेळेला the secondary oocyte undergoes second maturation division, meiosis second, equational division, but it is again very unequal, leading to the formation of two highly unequal cells. One is very much large, one is very small. One is very much large and one is very small. This larger cell is nothing but the oocyte. It is called oocyte. This oocyte, it grows into the ovum. That is nothing but the female gamete, ovum. And here this small cell form is called second polar body. Second polar body. या ठिकाणी ही सेकंड पोलार बॉडी तयार होते. अर्थात आपल्याला हे समजून घेतलं पाहिजे की सेकंड पोलार बॉडी इज फॉर्म ओनली आफ्टर द स्पर्म पेनिट्रेशन. Only after the sperm penetration, and that's why the formation of second polar body is an indication of fertilization. अर्थात सेकंड पोलार बॉडी केव्हा तयार होते? फक्त तेव्हाच ज्या वेळी स्पर्म हा सेकंडरी oocyteला पेनिट्रेट करतो. म्हणजे फर्टिलायझेशन कन्फर्म झालंय हे याच्यावरून कळतं जर त्या ठिकाणी सेकंड पोलार बॉडी तयार झाली असेल. आता आपण पाहिलं की हे सेकंडरी oocyteचं विभाजन होऊन एका बाजूला ओव्हम तयार होतं, फिमेल गॅमेट, एका बाजूला सेकंड पोलार बॉडी तयार होते. बट व्हॉट अबाउट दिस फर्स्ट पोलार बॉडी? दिस फर्स्ट पोलार बॉडी मे डिवाइड, I said may divide into two smaller cells again. May divide into two smaller cells again. That is two polar bodies again. Two polar bodies again. Okay? It means during oogenesis from single primary oocyte. Listen. During oogenesis from single primary oocyte, single ovum, single female gamete, and three polar bodies are formed. Three polar bodies are formed. Please try to distinguish between spermatogenesis and oogenesis. While discussing spermatogenesis, we have seen that in spermatogenesis from one primary spermatocyte, four sperms are formed. Isn't it? From one primary spermatocyte, मी स्पर्मॅटोजेनेसिस बाबत बोलतोय. स्पर्मॅटोजेनेसिस मध्ये एका डिप्लॉइड प्रायमरी स्पर्मॅटोसाइट पासून 4 स्पर्म्स तयार होतात आणि इथे मात्र oogenesis मध्ये फ्रॉम सिंगल प्रायमरी oocyte only one ovum is formed and 3 polar bodies are formed. Either 2 or 3 polar bodies are formed. In this manner, the ovum, the actual ovum is formed, and this is the entire process of oogenesis. आता आपल्याला शिकायचे that is the structure of ovum. Structure of ovum. But of course, we have to study actually the structure of secondary oocyte. कारण ह्या secondary oocyte मध्ये sperm penetrate झाला की फर्टिलायझेशन होऊन जातं आणि म्हणून आपण जे शिकणार आहोत ते प्रत्यक्ष ओव्हमचं स्ट्रक्चर नाहीये, ते प्रत्यक्ष स्ट्रक्चर असणार आहे सेकंडरी oocyteचं. सो नाउ लेट अस टॉक अबाउट द स्ट्रक्चर ऑफ ओव्हम इन सेकंडरी oocyte स्टेज. दैट्स स्ट्रक्चर ऑफ ओव्हम. That is egg cell or female gamete. And now let us see how is the structure of ovum. Actually, I told you it is not true ovum, isn't it? Whatever we are going to study, that is secondary oocyte, because yet the secondary oocyte not get converted into ovum. We have seen that secondary oocyte gets converted into ovum only after the process of sperm penetration during fertilization. So we can say that here we are going to study about the structure of secondary oocyte. Okay, now let us see. You know that ovum or egg cell is nothing but it is haploid non-motile female gamete. Non-motile female gamete carrying maternal characters into the offspring. Carrying maternal characters into the offspring. That is the characters of mother into the offspring. And the ovum is non-motile. So ovum, it is haploid non-motile female gamete carrying maternal characters into the offspring. The size of ovum in human being is near about 100 to 200 micron. 100 to 200 micron, or we can say in terms of millimeter 0.1 to 0.2 millimeter. 0.1 to 0.2 millimeter. Now let us see about the structure. Of course, the ovum is spherical in shape. Here I am going to draw diagram and simultaneously I will explain you. The ovum is somewhat spherical in shape. It is spherical in shape. It is spherical in shape. Ovum is haploid structure. Ovum is haploid structure. So obviously it encloses a single haploid and eccentric nucleus. Single haploid and eccentric nucleus. Nucleus is eccentric. Eccentric in the sense the nucleus is not present exactly at the center. Nucleus is present slightly away from the center, and this nucleus is haploid. Means what? It contains 23 pairs of chromosomes. 23 pairs of chromosomes. Which chromosomes? 22 plus X. 22 plus X. While studying sperms, we have seen that the sperm produced in human being are of two types. Half of the sperm with 22 plus X chromosome, half of the sperms with 22 plus Y chromosomes. Here the ova, all ova produced are of same type and having same number of chromosome, same type of chromosome, that is 22 plus X. Okay. Now here the nucleus is surrounded by cytoplasm. But here the cytoplasm of ovum is differentiated into peripheral and slightly thick egg cortex. It means the peripheral cytoplasm which is slightly thick, known as egg cortex. This peripheral cytoplasm also contains many tiny vesicles. Actually, we can say these are very small granules or tiny vesicles or very small granules. अति सूक्ष्म कण, लहान particles किंवा vesicles या ठिकाणी या egg cortex मध्ये present असतात. These tiny vesicles are known as cortical granules. What is the significance of these cortical granules? That we shall study in our next topic, that is fertilization. While inner cytoplasm is comparatively thin and known as ooplasm. Look here, with the help of slightly faint shade, white shade, I am going to show this is ooplasm. This ooplasm contains different cell organelles except centrioles. In mature ovum, the centrioles are absent and other cell organelles are found in this ooplasm. The plasma membrane of the egg cell, that is the thin covering of the ovum, plasma membrane of ovum is known as oolemma. Very simple. O, this word stands for egg or ovum, lemma stands for the covering. Oolemma, or it may be called as vitelline membrane. It may be called as vitelline membrane. Vitelline membrane. One point I want to add here. The cytoplasm of ovum, cytoplasm of human egg cell is free from yolk granule. Almost free from yolk granule. It means in the cytoplasm of ovum, yolk granules are almost absent. Almost absent. What are yolk? You know yolk? It is reserve food material. It is reserve food material. All of you have seen the eggs of hen, isn't it? When we break the outer shell of the egg of hen, you know some sticky whitish material comes out, that is albumin protein. That is albumin protein, and it is followed by a yellowish bulb, yellow colored bulb. That yellow colored bulb is nothing but the yolk. Means in that egg of hen, albumin and yolk, these are reserve food material. Reserve food material necessary for the growth of embryo, because you know in birds the entire growth of that embryo and development of young one takes place within that egg. So it requires large amount of food material, and there in that bird's egg that food material is in the form of albumin and yolk. But here the human egg cell does not contain yolk. There is no reserve food material, because you know the growth of embryo, growth of fetus in human being takes place in the uterus, in the womb of mother. आईच्या गर्भाशयामध्ये बाळाची वाढ होत असते, गर्भाची वाढ होत असते, आणि जे जे काही नरिशमेंट, न्यूट्रिशन आवश्यक आहे ते आईकडून पुरवलं जातं. आणि म्हणून या ठिकाणी ह्यूमन एग सेल मध्ये yolk हा रिझर्व फूड मटेरियल नसतं. अँड सच एग without yolk is called a lacethal yolk. A lacethal egg. A lacethal. A stands for absent. Lacethal stands for the yolk. Yolk is absent. According to some authors, according to some, we can say, scholars, there is a presence of little quantity of yolk, and hence the human egg is microlacethal. Micro stands for less amount, but truly speaking it is alacethal. That is, yolk granules are totally absent. Just I gave a reference of egg of hen, and in that egg, in all birds, large amount of yolk is present. That type of eggs are called telolacethal, megalacethal eggs. Leave that. But now this oolemma or this vitelline membrane, means this ovum, is further surrounded by two additional coverings. Further surrounded by two additional coverings, such as the inner one. The inner one is thin, non-cellular, made up of gelatinous material, jelly-like material, sticky material, known as zona pellucida. You have to remember these names. Zona pellucida. It means just outside the vitelline membrane there is a presence of non-cellular. Mind well, it is not made up of any cells. हा जो लेयर मी दाखवलाय झोना पेल्युसिडा हा सेल्युलर नाहीये, हा नॉन सेल्युलर आहे. इट इज मेड अप ऑफ जिलाटिनस मटेरियल नोन एज द झोना पेल्युसिडा. And again, this zona pellucida is also called as the primary egg membrane. For your additional knowledge, it may be called primary egg membrane. And zona pellucida is further surrounded by a layer of tall, column-like and compactly arranged cells. Tall, column-like, compactly arranged cells, radiating cells. These cells appear radiating away from the ovum. Okay, here I am going to show. This layer is called as corona radiata. This layer is called corona radiata. We have came across this term while studying Graafian follicle. झोना पेल्युसिडाच्या बाहेरून हा जो radiating tall column like cells आहेत या लेयरला म्हटलं जातं करोना रेडिएटा और इट मे बी कॉल्ड ॲज सेकंडरी एग मेम्ब्रेन. करोना रेडिएटा, remember these names, or secondary egg membrane. Here zona pellucida is called primary egg membrane and corona radiata is called secondary egg membrane. One more point, these cells of corona radiata are stuck with each other with the help of one material called as hyaluronic acid. In next topic you will come across these terms, so you should remember that the cells of corona radiata are stuck with each other, attached with each other firmly because of hyaluronic acid. Please remember these terms, hyaluronic acids. Now look at the diagram. There is a little space between vitelline membrane and zona pellucida. ही जी गॅप आहे या गॅपला म्हटलं जातं पेरी-व्हाइटेलाइन स्पेस. पेरी-व्हाइटेलाइन स्पेस. पेरी स्टँड्स फॉर सराउंडिंग. व्हाइटेलाइन मेम्ब्रेनच्या बाहेर असलेली स्पेस म्हणून तिला म्हटलं जातं पेरी-व्हाइटेलाइन स्पेस. अँड इन दिस पेरी-व्हाइटेलाइन स्पेस टुवर्ड्स वन एंड, या पेरी-व्हाइटेलाइन स्पेस मध्ये एका बाजूला जनरली the end towards which nucleus is present, जिकडे न्यूक्लियस आहे त्या बाजूला पेरी-व्हाइटेलाइन स्पेस मध्ये there is a presence of a small cell, a very small cell. Do you remember which this cell is? This cell is nothing but the first polar body. First polar body. We have studied few minutes before. थोडा वेळापूर्वी आपण शिकलो oogenesis शिकत असताना प्रायमरी oocyteचं मियोसिस फर्स्ट झाल्यामुळे दोन अनइक्वल सेल्स तयार होतात. लार्जर वन इज सेकंडरी oocyte and smaller one is first polar body. Now this first polar body is, we can say, present in this peri-vitelline space. Now this ovum, it shows polarity. It shows polarity means it has two poles, two ends. The pole or end towards which nucleus and first polar body is present, ज्या बाजूला nucleus आणि first polar body दिसते त्या पोलला म्हटलं जातं animal pole. And this opposite pole, त्याच्या विरुद्ध जे पोल आहे, that opposite pole is called as vegetal pole. Vegetal pole, just like the north and south poles of the earth, which are opposite to each other. So the pole or end towards which and first polar body is extruded is known as animal pole, and its opposite side, opposite end, opposite pole is known as the vegetal pole. Look, this is nothing but the ovum before fertilization, that is in secondary oocyte stage. Mind well. And that's why here in this peri-vitelline space only one polar body is seen. Only one polar body is seen. So here we have studied how is the structure of ovum. So let us stop here today. Thank you and take great care of yourself.